This video tutorial looks at transporting carbon dioxide, specifically the role of hemoglobin in transporting carbon dioxide and the changes in the dissociation curve at different carbon dioxide concentrations. Carbon dioxide is released from respiring tissues. It must be removed from these tissues and transported by the blood to the lungs for excretion. Carbon dioxide is transported in three ways. About 5% is dissolved directly in the plasma. About 10% is combined directly with hemoglobin to form a compound called carbaminohemoglobin. And about 85% is transported in the form of hydrogen carbonate ions, which is HCO3-. Carbon dioxide in the blood diffuses into the red blood cells. Here, it combines with water to form a weak acid called carbonic acid. This reaction, which is represented by number 2 on the diagram in front of you, is catalyzed by the enzyme carbonic anhydrase. So that's when CO2 is combined with water to produce H2CO3. The carbonic acid dissociates to form hydrogen ions, H+, and hydrogen carbonate ions, HCO3-, and that is represented by the equation 3 on the slide in front of you. The hydrogen carbonate ions diffuse out of the red blood cell into the plasma. The charge inside the red blood cell is maintained by the movement of chloride ions, so that's Cl-, from the plasma into the red blood cell. That is known as the chloride shift. The hydrogen ions building up in the red blood cell could cause the contents of the red blood cell to become very acidic. To prevent this, the hydrogen ions are taken out of solution by associating with hemoglobin to produce hemoglobic acid, which is shown in number 5 on the diagram in front of you, hemoglobic acid being represented as HB. The hemoglobin is acting as a buffer, a compound that maintains a constant pH. This diagram just shows that same series of reactions, but in a slightly different format. It's sometimes useful just to see it in different ways. You can see, for example, in the bottom right corner of this diagram, the chloride shift again. Blood entering respiring tissues carries oxygen as oxyhemoglobin. The partial pressure of oxygen in respiring tissues is lower than that in the lungs because oxygen has been used in respiration. As a result, the oxyhemoglobin begins to dissociate and release oxygen to the respiring tissues. This means that the hemoglobin is available to take up the hydrogen ions forming hemoglobic acid. Where the tissues are very active, there is more carbon dioxide released. This has a dramatic effect on the hemoglobin. The Bohr effect describes the effect that an increasing concentration of carbon dioxide has on the hemoglobin. Carbon dioxide enters the red blood cells forming carbonic acid, which dissociates to release hydrogen ions. These hydrogen ions affect the pH of the cytoplasm, making it more acidic. As with any protein, changes in pH can affect the tertiary structure of the hemoglobin and reduces the affinity of the hemoglobin for oxygen. The hemoglobin is unable to hold as much oxygen, and oxygen is released from the oxyhemoglobin to the tissues. Where tissues, such as contracting muscles, are respiring more, there will be more carbon dioxide. As a result, there will be more hydrogen ions produced in the red blood cells. This makes the oxyhemoglobin release more oxygen. So when carbon dioxide is present, hemoglobin becomes less saturated with oxygen. This is reflected in a change to the hemoglobin dissociation curve, which shifts downwards and to the right. And that shifting downwards and to the right is known as the Bohr shift. This Bohr effect results in more oxygen being released when carbon dioxide is produced in respiration. This is just what the muscles need for aerobic respiration to continue.